Well, good afternoon, everybody. Over 20 years worth of experience in microspheres, and very typically, on the right-hand side, what you see there is a homogenized sample of a microsphere with a very wide size distribution. What we've done is brought along a technology that gives you control over that and shows that you can get a monodisperse size at your size and choice of a distribution. And along the bottom shows the degree of control that we can have. By controlling the shear force, by controlling the flow rate, you can change the particle size. And that becomes important a little later in what I'm about to say. But before we do, what I'd like to do is to cast back to this conference in Boston in April.
So all of those downstream unit operations are important. They're important to control, they're important to not impact certain critical quality attributes of the product.
You know, with each different LNP, you're going to have differences in processes that you're going to have to make. So altogether, I would say the three main pieces of the downstream process are vital to the LNPs.
So we have there a statement of the problem where people at that stage in April were thinking about LNP manufacture as discrete unit operations. They weren't thinking of a whole process. What we have done since then, and what we are continuing to do, is think of LNP manufacture as a whole process. But let's start by looking at the traditional processes that are today. On the left-hand side, we've got the micromixing type of technique, which somebody said in Boston not very long ago at a vaccine conference, that it's not about scaling up, it's about scaling out in order to get to scale. And I show you a control curve, or control in inverted commas. I show you what happens at different flow rates with a T-mixer. Not a very controllable system, one would argue. What I bring to you, or what Micropore brings to you, is the AXF technology, the membrane-based technology. And not everybody walks around with a manufacturing plant in their pocket, but I do. This is what you are looking at on the screen. And it comprises a membrane inside a housing. In the top, you put your lipid. In the side, you put your RNA. Goes through the membrane, as shown there, and out of the end of it comes your LNP. Formed, but not yet stabilized. I bring you, based on that technology, I think, a world first. We have in front of you, and I apologise to the non-engineers, we have in front of you a complete process for manufacture of LNPs based on a single pass where you've got control all the way through. How do I come to do that? Well, we come from a manufacturing and engineering background. I'm a manufacturing guy, so I know what a manufacturing plant looks like. We start by saying, what does manufacturing really need to look like when you're thinking about LNPs? And then we start by going, how small can we go to get from that point to manufacturing scalably, reliably, repeatably, and robustly? And we'll return to this at the very end. So we start at the small scale with what we call our Pathfinder, our AXF Pathfinder. Just delivered to us and on test at the moment, debugging, but very much available. What we have here is something that will manufacture for you 400 microliters of sample into a 96 well plate. You don't have to use all 96 wells, but you've got a 96 well plate to play with. So it fits very well with some of the downstream stuff that we were hearing about a few minutes ago in terms of the analysis. It comes with two modes. The first mode, the small mode, is 400 microliters, and that will give you enough material to do your physical characterization and probably your in vitro testing. Switch the software over to develop mode, and then you will get the larger sample in a larger well plate, where you might be doing it with a 24 or a 48 well, larger samples, which will then give you the opportunity to do your in vivo testing. So you start out here with a set of formulation parameters, which you narrow in the discover mode to here, and then you finalize in the develop mode to here. And remember what I said earlier, what you develop on this is scalable to full scale. This is the intermediate version, the device I showed you a moment or two ago, where you can go from 1 to 200 litres an hour, and it's everything right throughout the range is fully GMP. The only replacement material are the O-rings. The membrane, sadly, is a very durable item. We have people in our lab who are still using the very first ones from 2016. So if I wanted to build a business model based on the razor and the razor blade, I've failed, clearly. What our process does, and I showed you the images earlier of the microspheres, this is what happens at the nanoscale. If we change the pore size and some of the other internal configuration, you can set a set point of the size that you want when you come to manufacture. Okay? So we've got our set point. Then if we control the flow rate on the right-hand side, you've got a lovely control curve. Compare with T-mixers. This is predictable. Under those circumstances, if you deviate from that set point for any reason, you can bring the flow rate up or down to get you back to the set point. And we were hearing earlier about control loops and being able to implement this in real time. We have. We can do this in real time and bring you a PAT control loop based on size. Does this stuff work? Well, yes, it does. Of course it does. Everybody's got the ability to do their encapsulation efficiency. It's a very gentle process. It runs at 2 bar, so it's very low pressure, no degradation. The in vitro dose response curve, exactly as you'd expect. And everybody's got their pictures of mice, haven't they? Well, here are a few of ours. They glow, they do things, they've got an IgG curve. It works. So back to this. What does this do for you? It gives you control. It removes variability. It gives you the potential of real-time release. And it gives you the ability to put PAT in at places, if I can control this, gives you the ability to put PAT in at the points you want. So as you have manufactured your LNPs, or as you have diluted them and stabilized them, we're working with somebody at the moment on a single pass TFF buffer exchange process. We're not quite ready to talk about that in detail yet, but I would expect to come back this time next year, and we would expect to be able to do so. And then, of course, it goes into sterile filtration. But you don't have to go sterile filtration with us, because going back to right at the very beginning, and the microsphere story, we're well used to dealing with materials that are way, way, way bigger than sterile filtration. We're well used to dealing with micron-sized materials. And therefore, you don't need, if you choose an appropriate feed system, if you choose to operate aseptically, you can choose the size of LNP or liposome or PNP that you want, and you are governed then by the therapy rather than by manufacturing constraint. When I showed this to the FDA in April, when I was over for the Boston visit, they loved it. They like the idea of continuous manufacturing because it removes variability. They like the idea of PAT because it gives you that real-time control that is irrefutably complete. And this is all Part 11 compliant. And by the way, we have just signed to sell our first 1 billion dose plant based on the first 2 stages of that. It's going east of here. Thank you very much.